So I'll get started. Again, I'm Jeremy Miller. I'm coming from the Allen Institute for Brain Science. Mike just gave a fairly good overview of the Allen Institute, so I will not talk about that very much, but what I would like to talk about is how we can find gene co-expression networks in the mouse, monkey, and human brain using mostly data from the Allen Institute. And I should say that most of what I'm talking about, because the monkey and human brain was done using more microarrays, and the mouse was done using more in situ hybridization. A lot of the networks were done in monkey and human, and then were validated as cross-species through mouse data. So to outline the talk, I'll first give a very brief introduction to previous WGCNA studies in brain. Then I will probably skip the brief introduction to the Allen Institute resources, or maybe talk about it for a minute or two. And then most of the talk will be about co-expression networks in the adult human brain, then how we can identify signatures of neurogenesis in the hippocampal subgranular zone in rodents and primates, and then talk about laminar and areal or regional specification in the developing human neocortex. So there have been a few studies of WGCNA in brain. Here's just 4 of them. One was identifying novel regulators of glioblastoma. So they found that that ASPM was a hub in a cell cycle module with several known cancer genes. Another found gene co-expression differences between human and chimp, and so there was weaker module conservation in the cortex than in subcortex, which makes sense because subcortex is a more, I guess, evolutionarily conserved region. They also found differential connectivity related to protein sequence divergence in this paper. There was another one identifying conserved modules in Alzheimer's disease and in normal aging. There we found that there's decreased expression in energy metabolism and synaptic plasticity modules. And there was also a potential role for oligodendrocyte dysfunction via presenilin 1. And there was the one that, another one that characterized the human brain transcriptome, and then WGCNA can be used to identify cell type markers from heterogeneous tissues, and this was from Oldham 2008. And there were modules for neurons, oligodendrocytes, astrocytes, microglia, and then there was a class of cells in the subventricular zone that they confirmed. And all of these references are available from the main co-expression network website. So those are previous studies of WGCNA. I also wanted to talk just briefly about a few things to consider when running WGCNA. Probably all of this slide will be review, but I thought it was useful to include in a single place. So first you want to talk about which probe should be used in the analysis. And as I've said before, you would typically use a single probe for each gene on the array using the collapse rows function. You can also filter genes beforehand, but that would depend on your analysis goal about whether or not you'd want to do that. Another question is which samples should you use when you're running an analysis. In most cases, the answer is all of them that aren't outliers, but it also depends on the question that you're being asked. Then you want to know, how do you choose your different parameters? I find that typically linear space works better than log2 space, but I know different people use both depending upon different things. And as was mentioned yesterday, WGCNA is very robust to your choice of parameters, and it's often fine to use the defaults. And another question is when you have when you want to cut your networks to choose modules, how many modules do you want to select? And I know Peter was saying yesterday there's different deep split parameters where you can choose a lot of modules or a small number of modules. And oftentimes when you have small modules, you can have very high confidence in your co-expression relationships, but if you have larger modules, you can have an easier time doing annotation because you'll have a bigger number of genes to choose from when you're doing your comparisons. And a lot of times I found that with few samples, it's better to use smaller number of modules, and I don't know if that's true for everyone, but that's what I found. Another question is, what can the modules tell you about biology? You can find out you can ask questions about what is known about the genes and how the patterns or the module eigengenes could relate to biology. Another thing you want to know is which parts of the network are preserved in other data sets, which we've already talked about in detail. And then I wanted to point out that visualizations are very important. And so things like Visant or Cytoscape can be very useful for displaying modules, and there's a lot of functions in the WGCNA library that will allow you to plot things in different ways. And in several cases, at least for these really complicated data sets that I'm going to talk about, we've had to come up with custom plots to fit the particular data set at hand. So this is background that you can look up on the PowerPoint after the fact when it'll be posted on the website. But I just wanted to sort of very briefly go over some useful things there. As Mike mentioned, there's a lot of different resources available at the Allen Institute website. And the three of them that I'm going to focus on are this adult human brain, where there was about over 3,000 arrays that were run in 6 human brains. And this one focused on breadth of coverage. So there's several hundred arrays run per brain. Another one here is the developing human brain, which is part of the BrainSpan project. And for this one, I'm going to focus on, there were over 1,000 arrays that were run on 4 prenatal brains spanning 15 to 21 post-conceptual weeks. And the analysis that I did focused on the cortical layers, so different layers of neocortex. And then there's another one, the non-human primate atlas, where there were over 500 arrays run in 5 brain regions that spanned different stages of the rhesus macaque development. And so those are the 3 main parts that I'm going to talk about. And also these mouse resources that Mike discussed in detail, we can use those to compare and contrast with primates, where we find networks in primate or human, and then see how the genes from these networks look in mouse. So the first one is this human brain atlas. So the question that you may want to ask with a data set like this is to start off with a single brain, which is what we initially had when we ran the project. If you have a bunch of samples across a single brain with all different kinds of, basically a lot of n=1s across a lot of different things, there are a lot of questions we can ask, like, are there certain genes that are specific to certain parts of the brain, like here compared with here? Which genes show similar expression patterns in the brain? Which regions show similar expression? So which regions show similar gene signatures? And how are the known markers for cell types distributed across the brain? And then do genes show different patterns at a global scale compared to if we only look at a very local smaller area of the brain? And so we can then add more brains, in this case a second brain. Do the gene expression signatures we find from the first one compare with what we find in the second one, and so on? And WGCNA is a good way to address a lot of these questions. So what we did initially is we created a network using all 911 samples from the first brain and ran WGCNA, and this is what the tree looks like, what the dendrogram looks like. We found that the genes clustered into 13 distinct modules, which can be seen in this top bar here. And a few of these modules correspond to known cell types. So what we did is, for this bar here, each of these colors represents genes that have been highly correlated with known markers for neurons here, oligodendrocytes, microglia, and astrocytes from other data sets. The earlier brain paper that I discussed very briefly before. And we found that in this data set as well, they do cluster together in individual modules. So that suggests that there's, some of these modules that we're picking up do correspond to different cell types in the brain. We also found good agreement with the second brain, which basically this is correlation across genes, across regions between brain 1 and brain 2, kind of like one of the plots that I showed in the previous lecture. And you can see basically all of these are red, which means there's good agreement between brains. And then the next thing we want to know is what we can learn about these different modules. This band here is module preservation. The more purple means the more preserved. A lot of them are purple, so a lot of these in addition to having gene expression that agrees between brains, we find that the modules are preserved between brains. And also, this here shows expression in the cortical areas versus all the other areas of the brain. And so some of this module here, these two seem to correspond to genes that have high expression in cortex, and these would have low expression in cortex relative to some or all other areas of the brain. And there's so many areas of the brain, you know, a 1 versus 1 comparison certainly is not all you can do, but it's a good place to get started. Let's see. So these plots represent the module eigengenes across 8 of the 13 modules we found. The bars represent genes that are samples that are from each of the different subregions, and the subregions are split into the various regions. So this is the frontal lobe, parietal lobe, temporal lobe, occipital lobe, and so on through different subcortical areas, and then cerebellum and white matter. And in some cases, there was more than one sample from a given subregion. I think there were about 150 subregions that were delineated, and so you can see there are error bars corresponding to that. And what you can see is, here's the sort of cortex network, or a module from before, and the expression tends to be higher over here than it does in many of these areas. This module corresponds to genes only enriched in the striatum and not anywhere else. And then there's the oligodendrocyte one has high expression in white matter, since oligodendrocytes also produce the myelin. So, and then there's other ones as well. But we can use basically some of these correspond to brain regions, some of these correspond to cell type. We find one of them related to translational machinery, so processes involved in different cells. And we can, as I mentioned before, we can use gene ontology, or we can use user list enrichment to come up with what these gene lists mean after we have them. On the right here are some of the hub genes that we found as being important for each of these modules, and just take my word for it that a lot of these hub genes are known in the process that I said. So like myelin oligodendrocyte, or actually I can't, MOG, I can't remember what it stands for right now, is the myelin producing gene, and it's in the oligodendrocyte module, which is what you would expect. And there's dramatic differences, for example, in many of these different modules, in particular between glia and neurons. So one question that we want to ask is, these all produce very general patterns. So the most different brain region is the striatum, and that's the one we found, and the cell types that are kind of all the way across the brain we found. But can we find things that are more specific to certain areas of the brain? There's only 20 or 30,000 genes, and can we use a different level of analysis to find different things using the same genes? And so we didn't find, in this case, modules corresponding to smaller brain areas like the dentate gyrus or individual midbrain nuclei, even though we know that there are markers for these. So what we did next is basically only look at one particular brain region. In this case, I'll talk about hippocampus, to see if the modules we find using WGCNA correspond to different things than they do when we look at all of the data together. And so in this case, when we use all of the genes and look only in the hippocampus, we find 13, in this case, coincidentally 13 modules that are different modules from the ones from before. These tend not to correspond to cell type. I don't know if you can see it from out there, but the colored bar genes here are spread out a little bit more. They're still very good between brain preservation and agreement. So what we're finding here does not appear to be the same as before, but it still is very similar between brains. So the question is, what do they represent? And so if we now plot the module eigengenes for some of these modules again, in this case, each bar represents a single sample ordered from different areas of the hippocampus and then rostral to caudal, or front to back within each area. You can see that these modules tend to correspond to patterns of expression within the hippocampus, both within and between subregions. So in this case, these are ones that are higher in CA3 and CA4. These are ones that are sort of increasing from rostral or caudally within regions and that are higher in CA1 and CA2. And so we can get more regional information if we look at only the subset of samples that are relevant to whatever region that we care about. And we found similar types of information when we look at other brain regions. So to summarize this part, we have used WGCNA to address several features of the human brain transcriptome. Across the whole brain, we find that genes group based on broad cell types and basic cellular functions and very distinct brain regions. But if we look across the hippocampus or other more local areas, we can find genes enriched in compartments and with rostrocaudal patterning as well. Neocortical regions tended to be enriched for the neuronal markers, and then there are certain subcortical regions that had more of the glial markers. And then we found that at the global scale, or the whole brain, genes tended in general to show slightly different patterns than at the local scale. So in this case, if you have this type of really complex data set, it may be useful to look on multiple kind of subsets of the data to try and find different things. And this type of analysis could be effective for any large-scale project that has a lot of different variables at once. So the next part that I wanted to talk about was the non-human primate, where, as I mentioned, there were, which I'll get into in a second, and we're going to use the mouse to compare and contrast some of these data. So this project, the idea was to look to see if we can find genes related to adult neurogenesis. And so neurogenesis, or neuron generation, was basically initially thought only to occur during early development and then to completely shut off as you get older. But there are at least 2 locations in the brain where this process is known to continue throughout adulthood. One is the subventricular zone, where cells generated along the lateral ventricle wall in mouse will migrate into the olfactory bulb and differentiate into interneurons. The other area that I'm going to focus on is the subgranular zone of the hippocampus, where basically throughout life, cells generated here will become dentate granular cells. And it's been shown that these new neurons that are generated in adulthood will make functional connections, so they're doing something. And the environment, or what's called the neurogenic niche, so all the stuff around these newly generated cells are also very important. Because if you take these precursor cells from these regions and transplant them somewhere else in the brain, they don't really show as much neurogenesis as they would if they were there. And if you transplant neural stem cells into these areas, they will generate into the correct kind of neurons. So there's something about the environment that's also important. And so this is a picture here of the hippocampus. This really, really dense band of granular cells is the dentate gyrus, and along the inside edge of this is the subgranular zone, and this is where this neurogenesis takes place. And so there are a lot of processes known, and a lot of the genes are known that are involved here, like when certain things are expressed and when they turn off and which types of cells are there. But this understanding of this process is far from complete. So we wanted to use these really large-scale databases at the Allen Institute to try and help address this question further. So the overview of this study was initially there was a comparison looking at the subgranular zone, or the inner part of the granular cell layer, versus the outer part of the granular cell layer in mouse. So this was used to find genes that were enriched in this layer. We then used some of the Allen Institute resources as well as PubMed to find more genes expressed in this subgranular zone. And then using the 20,000 genes on the Allen mouse brain atlas, we found about 350 genes that showed enriched expression in this particular region relative to other, relative to the overlying granular cell layer. And so this fourth part here is where we use WGCNA to look at data from the developing macaque monkey, which included samples from the granular cell layer and the subgranular zone at different points in time. So we can see if we can use this information from the developmental time course to kind of further refine what we learned from here. And then run some experimental validations of what we actually found as being important neurogenesis-related genes in this area. So the first thing we wanted to check is within the mouse data if the results that were found from the microarray experiment agreed with what we saw from the atlas. And this plot here shows basically so the things on the right are things that are genes that have higher expression in the subgranular zone, and on the left are ones that have higher expression in the granular cell layer. This black bar shows all of the genes that we did not find as being enriched in the granular cell layer in the atlas, and those basically are a perfect normal distribution around 0. But the ones that have that we found as having high expression in the subgranular zone through the in situ hybridization tended from the microarray to have higher expression in the subgranular zone as well. So that's the right shift of this red bar. So basically we're finding the same sorts of genes in both of our methods. And when we look at using gene ontology to see what these genes represent, we find canonical markers for neurogenesis, but we also find several markers for several different kinds of cell types. So for example, neurons, astrocytes, some types of interneurons, oligodendrocytes, and this is in line with the fact that there's a bunch of different things in that region, which is part of this neurogenic niche that's important for neurogenesis. So these are just some examples of the different patterns in mouse of the genes that were found. There's these astrocytes that have these different, you can't really see this very well, but there's kind of star-like scattered all across the region. There's interneurons, so these are zoom-ins. There's dividing cells, which there are very, very few of them, but all of them are in one or the other of the neurogenic regions. And then there's oligodendrocytes, there's blood cells, and then there was a bunch of different other genes that have this enrichment in this zone that we couldn't really tell which cell type they were from, but that previous studies in many cases had already pinpointed as being important in neurogenesis in some way. So more than likely, whatever cell types they're in, they're probably involved in this process in some way. And so the question is, can we identify more of these cross-species markers for neurogenesis using a primate model? And so that's where the non-human primate atlas comes in, where as part of the atlas, we have data from the granular cell layer of the hippocampus, the subgranular zone, and then the polymorphic layer, which is an underlying layer of the subgranular zone that has a lot of different interneuron and other cell types in there. And in non-human primate also, it has some of the important cells for progenitor cells as well. And so there are some species differences as well. This region in primate is a lot bigger than it is in mouse. And also, let's see. And so the question is, do we find common signatures in both species? And yes, and also another thing is that this subgranular zone is almost gone. There are still a few dividing cells in this area in primate, but the adult neurogenesis is not as extensive in primate as it is in mouse. And so the question is if we find common signatures in the two species. And so what we wanted to do is to cluster genes using WGCNA on all of the samples from the 4 time points. I forgot to mention what the 4 time points were. There's birth, there's 3 months, which corresponds to childhood. There's sort of juvenile and early adult, 12 and 48 months in the non-human primate. And so we wanted to use all the samples from those 4 time points in the granular cell layer and the subgranular zone as part of the analysis. And so when we ran this network, this WGCNA, we found a bunch of different modules corresponding to a lot of different things, but the ones that we wanted to focus on were the ones where the expression in the subgranular zone was higher than in the granular cell layer. And so that would represent the ones in red on this bar here. So these are the ones that should represent subsets of genes that may be interesting in the subgranular zone. And so we found that most of the genes that we found in the mouse, which are represented by these black lines here, did cluster in these 4 modules in the macaque, suggesting that these subgranular zone markers were common between species. The main thing that we found differently between these 4 different modules, and here are the module eigengenes. Here's the granule cell layer and the subgranular zone end from 0, 3, 12, and 48 months. The main thing different we find here is what happens with time in the subgranular zone. So this one, you can see a decrease in time in the subgranular zone. This one, at least between 0 and 3, there's an increase before there's a slight decrease. Here there's a big increase before a slight decrease. So basically what happens with time is what we see as being different between these regions. And so these labels were found from, again, using ontology and type enrichment analyses. And so the neurogenesis module, or corresponding to genes that are known to be related to neurogenesis, was the one we wanted to focus on. And so now we wanted to use phenotypic information to see, what this may represent, to get more confidence that this may be neurogenic related. So what we found, it was another paper that looked at quantifying neurogenesis by counting the number of genes, counting the number of cells in the dentate gyrus of monkeys of various age and seeing how many there were. And so that paper this was from a different paper, and they looked at, they found basically this is the number of cells in thousands from this paper at the 4 different time points. And this was the module eigengene expression of this module that we found. And you can see there's almost a perfect correlation in the number of cells that are proliferating at these different ages and the amount of module eigengene expression of this module. So that increases our confidence that this module actually is related to neurogenesis. Furthermore, when we look at the hub genes in this network, you can see the SOX11 and SOX4 are 2 of the hub genes, and these are canonical markers for neurogenesis, and they're required for neuronal differentiation. And so one of our collaborators, Nenad Sestan, in his lab, looked at what would happen if we knocked these out. They actually had knocked these out in cortex, but the cortex, it was a selective cortical knockout that also knocked out expression in the hippocampus. So we could take advantage of that experiment. And we found that when these were knocked out conditionally in the cortex and in the hippocampus, so this is a normal control hippocampus here, and so you can see expression in, I think, pyramidal cell layers here. When you knock it out, you end up with a mouse that has essentially no hippocampus. So whether this is related to adult or developmental neurogenesis, it still shows that the SOX4 and SOX11 are things that are critical for the development in the hippocampus. And it turns out that neither single knockout has an obvious phenotype, which suggests that these 2 genes probably have redundant functions. And there have been other studies that have also shown these genes have overlapping function. So we then wanted to look at the developing mouse brain atlas that Mike talked about from the Allen Institute, and see whether any of these genes that we found in our module, in our neurogenesis module, share similar time course of expression in mouse as they do in monkey. And what we found, so first of all, this is 2 of the genes in this module we actually had in situ hybridization expression data in macaque, as well as microarray. And you can see both for double coordinate and for this SEMA3C, you see the expected decrease in expression with time until there's very, very little at 48 months. And then in mouse, these same 2 genes, which are up here, if you start at this P14, so 2 weeks post-birth, you can see the slight decrease in expression with time, as well as with SOX11, SOX4, and these 2 other genes. They all seem to have a high expression here, lower expression here, and a lower expression here. So this suggests that, whatever sort of developmental process is happening early on goes till more like P14, which corresponds about to the birth time point in monkey, before you see the same sort of decrease with time. So overall, the subgranular zone neurogenic niche contains a complex combination of cell types, which includes radioglial and progenitors, dividing cells, immature neurons, astrocytes, vasculature, interneurons, so a bunch of different things. There's a high level of transcriptional similarity in this region in both mouse and macaque monkey, and then the makeup of the environment changes with development. So in particular, there's an expression of a group of genes that's highly related to the number of proliferating cells. And at least two of these genes have a known functional role in the hippocampal neurogenesis, and these two happen to be hub genes. So this is one example where if you identify the hub genes of a module, they are genes that are important for the process that you're looking at. So are there any questions about either of the first two parts so far? Okay. And so then the last thing that I want to talk about is the looking at the cortical layers and areas in the developing human brain. And so this is the developing human brain atlas. And again, we can use data from the developing mouse brain atlas to confirm things that we find. And so the subset of this data set that we're going to be looking at it comes from 4 brains at 15, 16, and 2 from 21 post-conceptual weeks. There's about 25 different areas from the neocortex, and within each of these areas, there are 9 different layers where laser microdissection was used to specifically separate out at both of these time points. So overall, there's about 500 arrays that encompass these 3 different axes. And so the brain develops in an inside-out fashion. So it starts off that you have basically a preplate, which then will become the subplate, and let's see, where is it? Subplate. And then once you have the subplate, the neurons sort of go past all of the ones that were already there and form this inside-out fashion. So at a given snapshot in time, what you'll find is that the neurons on the outer part of the cortex, the cortex are going to be older than the neurons going to be younger than the neurons below them because they will have just passed by. So when you're looking at the different layers during development, what you're seeing is both time as well as different layers of the brain. So, and interestingly, a lot of the layers, like most of these layers down here, and some of these layers up here are all transient layers that are pretty much gone by the time the brain develops. And so the 6-layer structure of the adult human brain only corresponds to about this much of the developing brain. But still, these transient layers are critical for proper development of the brain. And so another question is what makes the developing human or the primate neocortex different. And there's a lot of differences between the human or between the primate and the rodent. There's a very large secondary zone, the outer subventricular zone, which gets to be very big in human and is pretty much absent in mouse. There's also a very large subplate layer in human, and it's much smaller in mouse. And there's potentially some local generation of interneurons in human cortex, whereas in mouse, they're all known to come from somewhere else, and then they just end up there. And so one question we can ask with the data is, first of all, what's going on with human, and then is it the same and do the transcriptional things that we see in mouse recapitulate some of these differences that are known based on the anatomy? And so the first step was just to get a kind of an unbiased look at the data using multidimensional scaling. And so I can't remember if I think this uses all of the genes in the genome using all genes to group the samples together. And this is the 2 early time, the two earliest, the 15 and 16 week one, and these are the two 21 week ones. And what we can see is just in an unbiased way, this axis here, the first dimension, corresponds mostly to different layers of the brain. So in this case, these are ones that are postmitotic, so that are done dividing, and these are the ones that are in the germinal zone, so that can contain a lot of the proliferating cells. And then this other axis, which you can't really tell here, but it basically corresponds to things in the frontal cortex are up here, and things towards the caudal part of the brain are more towards the bottom. So in an unbiased way, we can see that there's a lot of structure in the transcriptome here. And so the question is, do we see these patterns using WGCNA? So one question is if we see these patterns using WGCNA, or if we can find other patterns as well. And so what we did is we first ran WGCNA on all 500, approximately 500 samples across all 4 of these brains, these in neocortex, and then just looked to see what we actually found as the different modules. So there were 42 different modules that we ended up with. There's definitely and so this bar here shows the change with age, so basically between the 15 and 16 week brains and the 21 week brains. And here are cortical plate versus subplate, and so basically genes that are higher in postmitotic versus proliferating cells. And we found a lot of different patterns, including a few that I wanted to talk about in detail. So these boxes here, these are the 4 brains, so 15, 16, 21, 21. And this box is these are the module eigengenes, where the average module eigengene expression for all of the regions in that layer of that brain are summarized by a box. So red means high expression and white means low expression. So in this case, this module includes genes that have high expression in the 3 germinal layers, slightly higher in the ventricular zone, and then have a slight decrease from the first 2 brains to the ones at the later time point. And then conversely, this one here has higher expression more in these postmitotic layers, and of the 4 brains, the lowest expression is in the earliest time point. So you can get an idea of both the layer and the time using this sort of a plot. What we don't see is anything about the region. It turns out that we didn't find any modules corresponding to different regions, probably because the different brains and the different layers had such a big effect that effect wasn't seen on this particular analysis, but I'll talk a little bit more about that later. And so one thing we wanted, and so one of these networks was, one of these modules was one corresponding to interneurons. And the pattern that we found here was an increase with time and high expression in these outer layers and in these lower layers. And for the most part, these are the expected layers that interneurons are found at this time. And this high expression in the ventricular zone is where it's hypothesized that these local generation of interneurons occurs in humans. So this might represent, something specific to human that you wouldn't find in mouse. But the 2 ones I wanted to talk about are this one, which includes genes that are expressed only in subplate, or highest in subplate, and then this one here that are the germinal layers. Okay, so the ones in subplate, as I mentioned before, that there's differences in subplate where it's much bigger in human than it is in mouse. And so one question could be whether these subplate genes are common between species, or whether there's different ones for the different species. And so these plots here show the expression of genes across the different layers and the different 4 brains. I've boxed the subplate, and so the further a dot is to the right, the higher the expression is in that layer. So there were several genes that showed high expression in subplate in both human and then in mouse. You can see these black bars, or these black bands here, which are basically expression in the subplate. On the other hand, there were also several genes which had no expression in subplate in mouse or expression somewhere else in mouse, but still showed high expression in the subplate in human. And conversely, there were some that showed expression in the subplate in mouse, but then were expressed in some other pattern in humans. So there does seem to be both an agreement between species as well as some that show very different patterns between species. And we used the WGCNA module, the hub genes in this module, as the starting point for input into the Allen Institute resources to look for the mouse genes. And so the other one I wanted to talk about is are the genes in these germinal zones. To look more in depth at this early time point and at these 3 layers, we repeated another WGCNA analysis, only looking at this set of the data and only looking at genes that were differentially expressed between the 3 different germinal layers. And the main reason for trying to put differentially expressed genes as the input into this WGCNA is because we really wanted to find genes related to the outer subventricular zone, which was the one that I was mentioning was specific to primate and wasn't really present in mouse. So we wanted to enrich our network for genes that could be in that particular layer. And what we found was there were 8 modules. 2 of them had much, much higher expression in the ventricular zone in both of these first 2 brains. So that would be, these are the module eigen genes shown in a heat map format. And I don't know if you can tell, but there's more red here and more green here. And same thing with these bars. So those are ones that have higher expression in this ventricular zone. There was one that had highest expression in the inner subventricular zone. And when we looked at the gene ontology for this, we actually compared this against a different previously published study of marker genes for intermediate progenitors, which are known to be expressed in that layer. They were mostly present in this module. So this module has the expected laminar expression here of the cell type that's known to be expressed there. And when we then looked at, and then most of the other ones had higher expression in this outer subventricular zone. But when we looked at what the enrichments were using gene ontology, we saw things like synaptic transmission and postsynaptic density. And so it's possible that these are actually genes that had high expression in the outer subventricular zone, but also had higher expression up here in the zones that are more related to neurons. But I mean, in general, we could find these modules that corresponded to the different layers and that were more specifically what we were looking for here in the different progenitor cell types. And we're actually following up now to see if we can experimentally show that some of these modules do correspond to different progenitor cell types, but that's still in the works. So as I mentioned before, we didn't find were genes related to aerial patterning or basically differences between region. And I think the main reason between this is that there were a lot of genes that we found which showed high expression in, say, the caudal part of the brain, but had much lower expression in the frontal part of the brain, or had high expression in the frontal part of the brain and then low expression in the caudal part of the brain. But almost all of these genes were this gradient that we found was specific to 1 or 2 layers. So it only represented a relatively small number of genes across a relatively small number of samples. So more than likely, these would have shown up as being modules that were smaller than the minimum module size that you would get using WGCNA. But in these 2 cases, we found the same pattern this is the ventricular zone for FGFR3, caudal enrichment in human. And when you look at mouse, in the germinal layers, you see enrichment over here, which is the same area. And conversely, the CBLN2 had high expression in the cortical plate in human. And then when you look at mouse, it's basically off here and on here. So you see, for a few of the genes, you find similar patterns across species, and the patterns you find are enriched for a certain layer of the brain. And interestingly, in human, there's a much bigger frontal cortex, and we actually found that more of the genes were enriched in the frontal cortex compared with the caudal in almost all the layers. So one could speculate that those two things are related. And so this sort of addresses the issue that in some cases you may want to do, in addition to your network analysis, you may need to do directed analyses if you have a really complicated data set and you're looking for some patterns that are not the most obvious patterns or the most variable things in your data set. So overall, we found that there's predominant gene expression variation were due to age and layer, and in particular the postmitotic or the layers versus the germinal layers, so the ones that are done that aren't going to be splitting anymore compared to the ones that are still dividing. Also, we find that genes cluster based on the expression in the layers and also in the main, in major cell classes. I don't remember if I talked about that, but we found some related to the major cell classes. We also found valuable information when we look at specific layers. So several distinct transcription signatures were in each of the germinal layers, and then we found similarities and differences between the mouse and the human subplates. And there are several genes which show layer-specific enrichment gradients, and I think there are too few genes and samples involved in these gradients to really find using WGCNA without using a really directed type of search. So overall, WGCNA is a very useful method for identifying patterns of co-expressed genes and for reducing the dimensionality of the data. We've been able to use it in several different projects related to the human brain, one looking at co-expression networks in the adult human, one looking at molecular signatures of the hippocampus subgranular zone, and also looking at laminar and cell type signatures in the prenatal human brain. But also it's important to be aware of the scope of the data set when using WGCNA. I'm not sure if this is relevant for a lot of projects, but when you have these really, really big data sets that we had, a lot of times you can find that signatures from local phenomena can be masked by much larger and also very important signatures. And also, if you have a large data set with a lot of replicates, so for example, if you have 100 samples from a single region, you can get networks that are very different than if you have the same size data set but that has a very different makeup. So for example, one sample from each of 100 regions, which is more of like what I was talking about here. So that's all I have. As Mike mentioned, these projects involve a lot of different people. Only these, and I only interact with a few people directly, but basically the projects involve a lot of people. So thank you.